Hello, highly welcome. This is Teacher Hillary, aka Prof. Hillary. You're highly welcome. Today we are focusing on still discussion on digestion. This is digestion part 2. Yeah, in our earlier episode, we had discussed digestion part 1, where we had discussed digestion in the mouth and in the stomach. Today, our focus is on digestion in the duodenum and in the ileum. Duodenum and ileum is small intestines. Duodenum is the upper part of the small intestines, while the ileum is the lower part of the small intestines. So, when food leaves the stomach, it gets to the duodenum. So, remember when food was getting to the stomach, it got there through a sphincter. It gets there through a sphincter. A sphincter is a muscle. So there is cardiac sphincter, which allows food to get to the stomach. As food leaves the stomach again, it goes through another muscle, another sphincter called pyloric sphincter. So as it leaves the stomach to the duodenum through the pyloric sphincter. Arrival of food in the duodenum stimulates the duodenal walls to secrete two hormones. The two hormones are one, secretin, and cholecystokinin. Pronounce that again. Cholecystokinin. Yes. So, hormone secretin stimulates the liver to secrete bile. Remember, bile is stored in gallbladder. So, liver secretes bile and it goes to the store. And the store is gallbladder. Hormone cholecystokinin will stimulate the store, gallbladder, to secrete bile. The same cholecystokinin also stimulates the pancreas to release pancreatic juice. Now, bile has some salts inside. Bile has salts. Sodium glycocholate. Or if you love it, say glycocholate, so that you don't miss the spelling for the sake of exam. Glycocholate and sodium taurocholate. Both of them are cholate. Glycocholate, taurocholate. Sodium glycocholate, sodium taurocholate. Those are bile salts. These bile salts are very important. One, it serves to neutralize the acidic chyme from the stomach. The food, remember from now henceforth, whether you have eaten veggies, sukuma wiki, and ugali, it is no longer ugali and sukuma wiki. It has been mixed in the stomach through the churning process. When it has been mixed together, it is no longer food it is no longer in parts. It is now one, and is some kind of a solution. It is now called chyme. So this chyme, as it leaves the stomach, it is acidic. Why do you think it is acidic when it leaves the stomach? Yes, it is because of hydrochloric acid. So, and the only part of alimentary canal which is acidic is the stomach because of HCl, hydrochloric acid. Otherwise, the entire part of alimentary canal is alkaline. So the salts, the sodium glycocholate, sodium taurocholate, or bile salts, neutralizes that acidic chyme to allow the enzymes in that region to act on food. Again, so it provides suitable alkaline medium for the enzymes to work. Okay, and now it neutralizes the acidic chyme. It also breaks down fats, a process known as emulsification. Digestion of fats is is a hard process, or breakdown of fats is a bit hard. So the fat droplets, the fats has to be broken down into tiny droplets to increase surface area for the enzyme lipase to act on it. The same way food is chewed in the mouth, that is mastication broken down, that is to increase surface area so that it can easily be acted upon by enzymes. Okay. Also, if said cholecystokinin stimulates the pancreas to release pancreatic juice, and this pancreatic juice has enzymes in it. It has pancreatic lipase, pancreatic amylase, it has trypsin, and it has another salt, sodium hydrogen carbonate. Let me handle one by one, or what it does. Starting with the pancreatic lipase. Lipase acts on lipids, fats and oils. Lipids are just fats and oils. So they break down lipids into fatty acids and glycerol. So that's the role of pancreatic lipase. Then pancreatic amylase will break down starch to maltose. You could be wondering, in the mouth, we had another enzyme called salivary amylase, which was doing the same work, breaking down starch to maltose. Why do we still have another enzyme in the pancreas known as pancreatic amylase doing the same work? Now, in the process of eating, you may eat very fast, and some of us have poor habits of taking water together with food. Sometimes the time, there is no sufficient time for the amylase to interact with this food in the mouth. So some of the starch may not be broken down to maltose in the mouth. The reason why we are fearfully and wonderfully made. God has another provision in this pancreas through pancreatic amylase to break down any remaining starch to maltose. The other enzyme is trypsin, which is secreted in an inactive form called trypsinogen. In our digestion part 1, there are other enzymes in the stomach which are being released in an inactive form. They are called their precursors, pepsin and renin. Now we have another third enzyme which is secreted in an inactive form, trypsinogen. The enzyme is trypsin. Do you still remember the reason why we gave for their secretion in the form that is not active? Yes. Yes. The reason is, if they were released in the form that in their active form, it finds that there is no food in the system, it can digest, it can self-digest. The body can self-digest, auto-digestion. It can break down the cells that produce them, because these enzymes break down proteins. Remember, our alimentary canal is protein. Si ni nyama tu. It is meat. Okay, so trypsin is secreted in an inactive form, trypsinogen, and then in the presence of food, it is now activated to trypsin by an enzyme enterokinase. Enzyme enterokinase. You can refer to episode 1 or digestion part 1 to confirm what was activating pepsinogen to pepsin and prorenin to renin. What was it? Hydrochloric acid, HCl. But now this one, trypsinogen to trypsin, is done by an enzyme called enterokinase. This time around, not HCl. You should know that. Okay. So that is duodenum, the upper part of the small intestines. Then the food goes to the lower part, the ileum. Arrival of food in the ileum stimulates the walls of the ileum, the epithelial cells, the cells lining the ileum, to secrete juice, another juice, intestinal juice, from the word intestine, intestinal juice, or it's also called Sacchus entericus, suckers entericus, or simply say intestinal juice. You don't have to bite your tongue as you as you pronounce suckers entericus or intestinal juice. This juice has some enzymes also. Now, you ask yourself, which enzymes are found in this juice? It is so simple. Now, how far is every component of food? How far has it been digested? To what extent? Now, you have the main three food components: carbohydrates, proteins. Okay, we have vitamins. We have also lipids. Remember, vitamins, we're not talking about vitamins in this case, in this digestion. Why? Vitamins are not digested, they are absorbed directly in the stomach. So far, how far is starch? Starch has been broken down to maltose. Maltose. What about proteins? To peptides. And lipids to fatty acids and glycerol. Okay, so to know the enzymes that are found in the digestive intestinal juice or succus entericus, you look at now. Ileum is where digestion is completed. So if it is where digestion is being where digestion is ending, then these foods should be in their final stage for absorption. Now, what should be the end product of carbohydrates? Yes, it should be glucose. What about proteins? At the end product should be amino acids. What about lipids? Fatty acids and glycerol. So we need enzymes that is going to transition, that is going to complete the process. So if carbohydrates is in maltose stage, if proteins are in peptide stage, so we need, for peptides to be converted to amino acids, we need an enzyme. The enzyme is called peptidase. For maltose to change to glucose, we need an enzyme, maltase. So which enzymes are found in the intestinal juice or succus entericus? So maltase, sucrase, peptidase, lipase, and lactase. So maltase is to break down maltose to glucose. Sucrose, if you have taken sucrose, said sucrose is found in foods such as sugarcane. Sucrose is a disaccharide. It is not, so it is not in its simplest form. So remember, food after digestion is in soluble form so that it can flow in the bloodstream. So sucrose should be broken down. So we have sucrase enzyme that breaks down sucrose into fructose and glucose. We also have peptidase, which will now break down peptides to amino acids, the simplest form of proteins. And lipase is to break down any remaining lipids to fatty acids and glycerol. Now, so the food at this point, the food is now a solution and soluble. It is no longer called chyme. When it left the stomach, it was called chyme. When it has now been broken down to the simplest form, soluble, it is no longer called chyme, it is now chyle. Same total, chyle. Yes, CHYLE. This is now a soluble product of digestion, ready for absorption. What is absorption? The process by which it passes through the cellular lining of the small intestine, villi, into the bloodstream. But now go to the cells, broken down to release energy. Amino acids are used to build proteins, make enzymes and hormones, which will build our body. So that is digestion part 2. Thank you so much for joining, and continue joining and at YouTube, Prof. Hilary, and we can always meet there for more discussions. Goodbye.